Hi everyone, it's my pleasure to be here. My name's Tom Forbes. I'm a paediatric nephrologist. I work down at the Royal Children's Hospital in Melbourne and that currently occupies around 10 to 20% of my time. About 2 years ago I started a PhD looking at stem cell biology, and that currently occupies about 150% of my time. So all up I'm around 170, 180% EFT I think. And the people who helped me with this project are illustrated here, and I thought I'd just give them acknowledgement straight up. So we're the Kidney Development, Disease and Regeneration Group at the Murdoch Children's Research Institute in Melbourne. And that's a group that's led by Melissa Little, who's a very highly esteemed renal developmental biologist and stem cell expert internationally. So I'm very grateful to have her supervision. But everybody here on this slide has contributed to the work that I'm presenting today. So that's fantastic. So I thought I'd really take it right back to basics and start with what is a stem cell. A stem cell is a cell basically that within an organism can become any other cell type in that organism, whether that be a mouse or a human. And that's illustrated here on the, have I got a pointer? Yes, illustrated here in this image on the left. And what you can see is that the stem cell up here at the top can take numerous different paths down this theoretical landscape where it will be guided by its environment to certain types of cells. And that could be a kidney cell or a liver cell or a little toe cell or an eyelash follicle cell or whatever you like. And the other nice thing about stem cells is that they are self-renewing. So what I've shown here is that we can put some stem cells in a plate, and after a couple of days they will have multiplied themselves numerous times, and we'll have heaps of them, to the point where we need to pick them up, shake them around a bit, and put a proportion of those cells back into a new plate. And that's great because it means that we don't have to go back to the primary source of where we got the cells from in the first place to be able to keep using them for serial experiments. Once we've got stem cells from somewhere, we can keep them not indefinitely, but for pretty long periods of time. And so we've known about stem cells for about 20 years, and for the first 10 years, the only way that we could get human stem cells was actually to obtain them from discarded fertilised embryos from in vitro fertilisation. And that was obviously a contentious ethical problem at the time. But around 10 years ago, two very intelligent Japanese scientists by the name of Takahashi and Yamanaka published some work that won them a Nobel Prize, and that was induced pluripotent stem cells. What they did was to take skin cells from humans or from mice, and they forced the expression of a few genes inside those cells, which really took over the cell and turned it back into a stem cell definitively. And once that's done, the stem cell becomes a stem cell and remains a stem cell if it's cultured under the right conditions. And from that point, people have started to develop protocols to turn those stem cells into various types of organs. And as long as you can identify the cues in a normal human developing embryo that will cause a stem cell to turn into that cell type, you can replicate that in a dish. And about 2 or 3 years ago now, Professor Little and her laboratory, prior to my joining the lab, published this landmark study publishing a protocol to turn stem cells into kidney cells in a dish. And this is the cover of the journal that was published in, and one of the kidney organoids that we've cultured in the lab. And you can see they're only small. That's a 1 millimetre mark there, so it's only sort of 2 or 3 millimetres wide, but you can see all the tiny little structures here. And so we call this an organoid. And an organoid is basically a three-dimensional replication of a human or animal organ in a dish that's miniaturized but still mimics all of the normal microanatomy of that organ. And this is how we do it. So on day 0 over here, we will put the stem cells into a culture dish, and then we'll start washing those stem cells with media to both keep them alive, but also give them some growth factors or various compounds that mimic what a stem cell would see in a normal human on its way to becoming a kidney. We're just really recapitulating that process in a dish. And so there are two growth factors that we use in this protocol. One's called CHI and one's called FGF9, but that's beside the by. And we do that for 7 days, and we get to a point where we can then lift up those cells, take them off the plate, shake them up into a single cell suspension, and then spin down numbers of cells in tiny little tubes. And we centrifuge them at high speed, and that compresses all of those cells into a little pellet. We can then pick up that pellet and put it onto some mesh, and that mesh can keep culturing. And for the next 2 to 3 weeks, those cells then reorganise themselves in that pellet and turn into little nephron structures, as Shilpa was illustrating to you before. And so this is what they look like. This is the transwell dish. So this is the mesh here that you can see. And then here you've got 3 little organoids growing about 3 millimetres in size. And this is again that more high-powered view of the organoid. And if we then stain these tissues with little flags that show us what the cell types are, we can actually see the individual cell types of the nephrons inside these regenerated organoids. So the red here are the glomeruli, or the glomerulus that Shilpa was talking about before. And from those little red glomeruli, you can see blue tubes, project all that well in here, green tubes, and then little pink tubes. And they're the 3 different segments of each nephron that are all in continuity in these little organoids. As Shilpa had suggested, there are about 25 different cell types in the kidneys, and we've characterised more than just these 4 cell types. We have the support structures, we have some early cells that may contribute to the development of blood vessels in these organoids, but what we don't have is any frank blood supply, and we don't have a urine drainage system, and obviously these organoids are not making any urine in the dish because they don't have that blood supply or that pressure. So now we sort of find ourselves in the situation where we can take a skin cell, and actually now we can do this from a blood cell as well, so we can take a blood test and generate the stem cells from a blood test. So it's a very non-invasive process. But we can take those cells, turn them into stem cells, and then turn those stem cells into little mini kidneys, and we can use them now to model disease, test drugs, and potentially in the future generate therapies that will be of direct benefit back to that patient. And I've separated these applications out into things that, in my opinion, are reasonable expectations over the next 5 years or so, and those that have got some feasibility work in process that has not been as successful as perhaps we might have wanted it to be. But I thought I'd mention them anyway.
As you go along, can you discuss the artificial kidney?
I will. I'll get there at the end. Unfortunately, that's one in the latter group. So disease modelling is what I'm doing my PhD on, and this is what I think is going to be the sort of immediately realisable application of these organoids for the treatment of kidney diseases. And disease modelling really just describes the process of recapitulating a disease or creating a disease that's in a dish that you can then study, that's an exact replica of the disease as it would occur in a person. And previously all we had to go by for disease modelling was using mouse models or fish models or just flat 2-dimensional cell culture systems, which I mean, I don't need to tell you, it's quite intuitive that there are limitations to what can be extracted from that sort of research and directly applied to human disease. So what we've got within the organoids is a 3-dimensional tissue. It's human in its genetic origin. In some cases, it's patient in its genetic origin. So I can look at the exact whole patient genome in the organoid that I'm studying, including both a gene that is causative of disease and any other unknown genes within that set of DNA that might be altering how that disease is working. And also we have the opportunity to make as many of these as we want just from one blood test. And that gives us the opportunity to do high content screening, and I'll explain a bit more about that in a minute. I thought it was really clear to talk about, really a good idea to talk about the types of disease that we can model with these organoids, and genetic kidney diseases are by far and away the top of that list. And there's a lot of people around the world working on these genetic kidney diseases in the organoids at the moment. And also medication-related kidney toxicity, we're getting close to working on that, and I'll mention that in a minute. But there are some things that the organoids are not ready to model, and those include inflammatory disorders and vascular kidney diseases like lupus or ANCA vasculitis, And that's because these organoids don't have blood vessels, they don't have immune systems, so it's difficult to recapitulate those diseases where the body's not working very well and the kidney's a bit of an innocent bystander. Congenital abnormalities of the urinary tract. So these organoids don't have a ureter. The urine doesn't get drained from a tube into the bladder, so we can't really model those diseases. And then there's a bit of a question mark over adult onset kidney diseases, genetic kidney diseases, or acquired kidney diseases like diabetic kidney disease or ischemic kidney disease. And that's because when we look at the gene activity profiles of these organoids, they are roughly similar to a trimester 1, trimester 2 fetal kidney. So if you compare the gene activities to a range of different types of human tissues, they marry up most closely to fetal kidneys. So we're really talking about, at the moment, modelling early onset kidney disease. But there are certainly a lot of people working on trying to improve the maturity of the organoids. And there are some organoid structures from other types of organs, like brain organoids, for example, where people are modelling Alzheimer's disease. So I don't think it's out of the question to think that one day we'll be modelling adult onset kidney diseases with these models. So I wanted to just give you a quick case report on one of the stories that we've published recently. And this was an 11-year-old girl who was diagnosed with a retinal disease, an eye disease when she was 1, and then end-stage kidney disease when she was 5. And she's subsequently been transplanted, received a kidney from her father. She had a novel next-generation genetic sequencing test which found that she had 2 mutations in IFT140, which is the name of the gene that causes her disease. And what IFT140 does, this is a very quick science lesson, from every cell in your body you have this little hair, a little hair that sticks up from the top of the cell. It's like a bit of an antenna for the cell, and it senses what's going on around the cell and tells the cell what's going on. And it's called a primary cilium. And IFT140 is a gene or a protein that drags things out of this cilium and back into the cell. So it's really responsible for getting stuff out of the cilium and putting it back into the cell. And so when it doesn't work very well, you get this sort of baseball-shaped primary cilium where it's all swollen at the top because you can't get that stuff out of that little antenna. And so it develops this abnormal shape. What we did was take this girl's skin cells and we turned them into stem cells, but at the same time we used what you may have heard of called CRISPR, which is a new way of editing DNA, and we were actually able to fix her gene mutation in her own cells so that we then had 2 sets of stem cells. One set of stem cells that perfectly replicated her genetic disease, and one set of stem cells that for all intents and purposes was still her, but her gene mutation was fixed. And we were able to compare the two. And we made kidney organoids out of both the stem cells, and we looked at the shape of those little hairs sticking up out of the cells. And what you can clearly see is that we've got the baseball-shaped primary cilium, the baseball-shaped antenna in the patient organoids, but that the gene correction has fixed that, and we've got nice, normal, straight little cellular antennae in the gene-corrected organoids, and we were able to quantify that. So this gave us 2 pieces of information. One, it validated that when you make a kidney organoid from a patient's stem cells, it will have that patient's disease. If it's a genetic disease, it will have that patient's genetic disease in the organoid, so we can study it. And this is another thing that I wanted to just show you. This is a video of a bioprinter that we've obtained in our laboratory in collaboration with a US-based group called Organovo. And at the moment what the bioprinter is doing for us is literally just printing the organoids, so replacing me, which is fine because it takes an awful long time to make the organoids, and it probably takes about 3 or 4 hours to make a whole plate of organoids in the lab, and this machine can do it in about a minute. So that's nice. It gives me time to focus my efforts on other things. But the other thing that this allows us to do is to make organoids in a high throughput manner. So what that means is making hundreds of organoids from a patient line in multi-well plates. And once you've got them sitting in the in the plates, you can then screen drugs. So we might want to screen drugs that are already being used in asthma or epilepsy or things that nobody had ever thought of. And we can just give them a go on the organoid. And as long as we've got a readout from the organoid that tells us what the disease activity is, we can start to say, all right, 999 of them didn't work, but that one has done something. Let's look further into that. And this is the mechanism by which we're going to start to develop new medication treatments and repurposed medications for drugs that are already widely and cheaply available on the PBS that might have an effect on genetic kidney diseases. And then, in answer to your question, these are some of the more aspirational applications of stem cells, which speak more to the cellular therapies. Can we use these cells and put them back into a patient to correct kidney disease? That would be of benefit from a transplantation point of view because, of course, if you're putting someone's own cells back into their body, there's not going to be a need for immunosuppression, one would expect, because there's not going to be any poorly matched cells in that therapy. So there are a few different ways that might occur. One is just injecting the cells as therapy, and there have been a few attempts at this using human kidney stem cells into mice that have had a kidney injury. And there's been a documentation there that you can see this red tubule here is actually a human tubule that's sort of been incorporated into the healing process of that mouse's kidney. So there's the possibility that we might be able to add cells back into an injured kidney and have those cells help with the repair process. That's at a very early stage, and one of the key issues with that is how do you get the cells back in? Because you can't just, this particular study just injected them into the vein of the mouse, and I can tell you that the vast majority of those cells are going to get stuck in the lungs and die. And so the only way to really get those cells where you want them is to put them into the renal artery, which is quite an invasive process, especially for a patient who's got an acute kidney illness. So that's something that's in the very early stages. This cellular assisted dialysis is also a bit sort of conceptual at the moment. For those of you who might be on dialysis, you'll know that you get hooked up to a machine and your blood gets passed through this sort of plastic chamber where the solutes get moved across the little plastic wires. And what we're talking about here is exactly the same thing, but with cells, kidney cells lining those tubules. So a little chamber that's full of cells that are purpose-built for cleaning blood, and whether that may be an adjunctive little chamber to the chamber you already have to improve the clearance offered by dialysis. And then finally, this is the concept of scaffold recellularization. And what that means is taking, for example, an animal kidney like a pig kidney, or maybe a donated human kidney that hasn't been allocated anywhere. And what we can do is actually wash out all the cells from that kidney and just leave behind the scaffold. So there's no cellular material and just leave behind the scaffold. We're very good at doing that. What we're not very good is actually managing to repopulate that scaffold with the stem cells. Many people have had a go. We can get the cells part the way, but as you can imagine, all of the avenues for the cells to get into the depths of the kidney are absolutely tiny, and you can't just push the cells up those little avenues. So that's very tricky. And so that's all of these things are concepts that may become relevant in future, but there's a fair bit of work to be done. So in summary, what it's important to understand is that induced pluripotent stem cells can now be derived from any individual, any organism. We could do it from your cat, we could do it from any individual. And that's something that can be done relatively easily by many labs around the world. And of course, the genetic engineering is there now to fix gene editing errors. Kidney organoids can be cultured from the stem cells, and it's likely that we're going to start to develop new treatments for stem cells because of the high throughput nature we're going to be able to generate to study new treatments. In the longer term, I think cell-based therapies are a bit far off, but they might mature to the point of suitability for clinical use at some stage. And on the first slide, I put my Twitter handle, which is @KidneyTom. And so if you want to keep up to date with what's going on in the research space around kidneys and stem cells, you're more than welcome to follow me. Don't look too closely at my profile pic, because as I realised this morning, I think I'm wearing exactly the same clothing today as I was in my Twitter profile pic, and that might be a marker of the austerity measures I'm living under while I do the PhD. But you're more than welcome to follow me, and thanks very much.
Do we have any questions from the audience? Do we have any online? Yeah. Any questions? Not yet? Yeah, hang on. If we have our hands up, and I'll bring over this so the online community can hear. Great. Michael.
Thanks very much. Yes, just the cellular assisted dialysis. You said that's primarily conceptual at the moment. How primarily conceptual and how practical?
Yeah, so the trick here is to choose what cell type we want, because the kidney tubule itself is made up of multiple different cell types as you move down from the glomerulus, the filtration unit, all the way down to the bladder. It's completely different cell types. And so which cell type do we want? Does it need to be patient-derived? We don't know, because if you start to wash your blood through somebody else's cells, will you set off the immune system? We're not sure. What do we want the cells to do? Do we want them to just clear salt? How do we stop them from spilling protein? And then I think after we've worked all of that out and actually designed the chamber, I think the question then becomes, what are the potential side effects of washing your blood over a cellular surface? And will that start to activate and aggravate problems that we know can occur in dialysis, like infection and blood clots and those sorts of things? We haven't yet seen a bioartificial chamber that has been lined with the kidney cells that's been used. So it's something that's been talked about, but it's a fair way off.